Okay, next up, I'm pleased to welcome Geoff von Maltzahn. This is somebody that I've wanted to hear from for a long time. He's a partner at Flagship Pioneering. And you might ask, what is a Flagship Pioneering? Well, it's not a venture capital firm. They do more than that. They're located here in Boston. They actually start companies, build them from the ground up in cutting-edge areas of biotech. They're best known for starting Moderna, maker of the COVID-19 vaccine. And Geoff works there, and he's actually started, he tells me, 10 companies, maybe more, and has another 6 in stealth. But he's going to be talking about one of the most important ones, which is called Generate Biomedicines. And it's just a very clear example of how AI, the same type of AI that's in ChatGPT, how AI is being applied to drug discovery. So it's great to have Geoff here. He's also an MIT grad, class of '03, and has a PhD from MIT as well. So if I can call Geoff forth onto the stage.
Thank you, Antonio. Yep. So as Antonio just said, I was once an undergraduate in this audience, so it's beautiful and intimidating to be standing up here presenting some of the stuff that I'm working on to you. So as both of them just hinted at, if you could imagine some beautiful tapestry behind the screen here, and there was the history of all of life sciences on it, maybe all of material science, and maybe all of science, and you could wander up here and stick your finger on when you would have wanted to live, that tapestry would be worn through on right now. And it might still be the case if we had access to the decades to come. And the reason for that is that the world that we live in and have has been a product of human ingenuity discovering and engineering things. And now the river of human intelligence has the river of machine intelligence flowing into it. And a lot of the technologies to come are going to be generated, not discovered or engineered. And the implications of that for biology are amazing. I mean, there isn't a human brain on the planet that could glance at this slide and tell you whether any one of these paragraphs would cure you, kill you, or win you a Nobel Prize. But of course, what paragraphs do in biology is they can encode, a cascade of information whereby a sequence of DNA becomes a strand of mRNA, becomes a sequence of amino acids, which then folds itself into a technology we call a protein. In fact, sometimes when I tell relatives about the work we're doing in Generate, they think protein might mean bacon at first glance. It's sort of unfortunate that we use the same word to describe the most amazing and often smallest sensors, machines, motors, and devices, proteins, and soy, etc. So for the purpose of this talk, imagine the former is what I'm describing, and this feat is the equivalent of this building, your car, your iPhone, arriving at your house as a noodle and folding itself into the technology that you enjoy. I mean, this is beautiful, and of course, this is why life is so magical. And implicitly, all of the information of the quantitative function of that protein must be present in the DNA sequence from which it arises. So about 7 years ago, we started to ask, what if we could smash those two together? And in fact, learn not just to see DNA, which is what the sequencing revolution has allowed us to do, but in fact, read what a sequence of DNA means in terms of quantitative function, or potentially go in the other direction, which is to desire a function and know what sequence of DNA to write. And if we could do that, biotech changes forever. I mean, the little animation that you just saw here would be enabled by asking, what is the DNA sequence of an antibody that would stick here, 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 or here? And today we haven't had tools to do anything resembling that. But of course, because, for example, predicting what an antibody would need to look like to bind to a given location would be very valuable, it's had 5 decades of attention, and most of that has been with the application of physics. So 6 years ago, we thought it wise to potentially depart from that tradition and ask whether some of the advances taking place in machine learning might be applicable. And in particular, whether advances taking place in application of machine learning to images might be applicable to this. And this is a beautiful New York Times article from a couple of years ago highlighting generated individuals. None of these people have ever existed, and statistically, they won't ever exist. And these are generated by models that have learned nothing explicit about human hair physics, human eyeglasses physics, or skin, but they've mastered the rules of what it means to be a human face so as to generate uncountable numbers of new examples. As you can see, that's quite distinct from just facial recognition and categorizing things that already exist. And we wondered, well, perhaps the same could be true in biology. Maybe if we could grab onto every genome that has been sequenced and made publicly available, there would be relationships between the language of DNA and the quantitative function of the proteins that flow from it. And that if you were going to pick one function to focus on in biology, this is one of these rare moments where it's easy to be disciplined. All antibody value, all peptide value, and nearly all cytokine growth factors, et cetera, have one function. They bind to a target in a specific location while not sticking to other things. So maybe the rules by which a protein would like to bind to a target might be inferred from the way they do like to interact with one another inside of proteins. So we were able to pull two rarities in biology, large, very high-quality data sets from genome sequences and from all of the three-dimensional crystal structures that have been elucidated over the past few decades to start to learn how to generate, not categorize them, but start to ask questions of what would the DNA sequence of a protein need to be to embody this function? And in particular, a function like what would the sequence of a protein need to be to bind this target in this orientation at this location? And publicly available data sort of allowed us to start to crawl. We had to build a platform that would allow us to test very large numbers of generated sequences that our models are proposing would be capable of embodying a given function, build them and evaluate them in the lab. Thus far, we've been able to do that for now millions of sequences that we have generated and tested. And one of the beautiful things about machine intelligence sitting in the center of one's endeavors is that every rep, with every failure, in fact, with enormous numbers of failures, the models get wiser. And whereas we would zoom in on the few things that work, ignore the rest, the full range of spectacular success and spectacular failure can benefit machines. So I'm going to describe to you some of the things that to us have been remarkable that have come out of those models. So we've been able to generate proteins that have the ability to bind to epitopes that, as far as we're aware, other antibodies haven't bound to previously. We have examples of being able to bind to targets that have been bound, but to embody superior therapeutic characteristics in the molecules that we create. And I'll give you a conceptual example that illustrates some of the advantages of these methodologies. So when I was still CEO of Generate, we challenged the team to take the top $50 billion of antibody therapeutic sales and generate antibodies that would hit the same targets in the same epitope, in the same binding pose, with the same structural conformation at the interface, with comparable or more favorable affinity, without going near the parent intellectual property of the starting molecule. Within 3 months, the team was able to do that for all of them. And to put that in comparison, if you had a big biotech company's resources and team, you probably wouldn't be able to do that in 5 to 10 years with traditional approaches. What I just described is like Robin Hood splitting the first arrow with a second one. You'd be able to find stuff that sticks elsewhere, but not fulfill those criteria. So that offers the possibility of, when there's a therapeutic demonstration, enabling the best drug to be provided to patients for that. This is one of my favorites, which is we've been able to recode enzymes. There's a handful of bacterial enzymes that are used in medicine today. As you can imagine, our immune systems don't like that and rapidly generate neutralizing antibodies against them. we asked whether we might be able to rebuild one of those enzymes so it would be composed entirely of peptides that are already in us or are difficult for our immune system to present and recognize. To do that, we had to change more than 100 amino acids and do it simultaneously. To put that in perspective, every time you change an amino acid, you have 19 options. So that's 19 to the 100 power. That is atoms of the universe squared combinatorial realm. And with sort of like a coin flip, those sequences would in fact function like that parent enzyme. And when we were predicting up a functional gradient, some of them had a higher catalytic efficiency than the parent enzyme. The same models that are able to engineer antibodies have the ability to generate peptides. We've started to apply this to cell therapies and more. And this is one of the applications that we are applying this to. This is nobody's friend, SARS-CoV-2. What you're seeing here is the spike protein doing its thing. It has two domains. One that's like Velcro, that binds to ACE2. The other that performs that miracle, which you just saw, which is acting as like a vice to force two membranes together and allow the virus to deposit its contents into our cells. Unfortunately, our immune systems aren't good at finding antibodies against that second portion. And the reason that's unfortunate is while the ACE2 binding domain, the Velcro, has changed rapidly since the pandemic began, this domain, probably because it is so dynamic, hasn't changed almost at all. But we've been able to generate antibodies that can hit this domain that we hope will be capable of preventing future variants and preventing the whole suite of variants that we have observed thus far. And as best we know, this is or among the first generative AI-enabled antibodies to move into the clinic. So I want to leave you with one thought, which is that sometimes it's tempting to think that everything in nature has been optimized because Mother Nature's had her lab open for a really long time. And that is true. But if you take an average size protein, you've got 100 plus options, 100 plus amino acids and 20 options each. So going back to the math, that's 20 to the 100 plus power. So even with generous assumptions around everything Mother Nature has ever done, she has only been able to test the equivalent of one drop of water versus all of the Earth's ocean of combinatorial potential. So everyone in this room, everyone we know and love, all the biology that we have ever observed is in that one drop of water. And so what if tools like these can allow us to sail the seas, not just incrementally, or even convert the seas into another dimensional realm where function clusters together instead of being scattered about a random sequence realm? I think the opportunity for this is extraordinary, and my finger is on right now for when to live as a biologist. Thank you for the time.
Thanks, Geoff. That was a nice talk.
Thank you.
Again, if you have questions, put them in the app. Something Geoff didn't mention is that Generate, a company that he is chairman of, has raised close to $700 million. This is a titanic sum, as long as we're on the navigational metaphors, titanic amount of money. Why does this company that is doing generative AI for protein design, why does it need so much money right now?
Yeah, great question. Well, so I'm not chairman of Generate, but I'm a co-founder and on the board of Generate.
Oh, okay.
The reality is it's difficult to create a therapeutic company that can bring more than one thing into the clinic with less than $100 million, particularly if you're starting from scratch with a new technology. If you're translating something, you can do so less expensively. And if you believe that a technology has applications that are broad, like I just described, it'd be a big mistake to bring one thing into the clinic. You know, the simplest heuristic to build one strategy with would be to, on any given time scale, maximize the probability of building the company that you really want to build. The one we really want to build is one that gets to change medicine. And it'd be nice if that didn't require a lot of capital, but the reality is...
Okay. What, look ahead 10 years. What will the company have done?
Well, I'll start with the field. So that combinatorial math that I was doing, which arrives at atoms of the universe, you can arrive at that for almost any protein. And antibodies are no exception. So just the binding domain of an antibody has atoms of the universe combinatorial space. And when you or I are infected with something, unfortunately, our B cells don't have access to all the atoms of the universe to discover a solution. And it's more amazing that we ever find a good answer than it would be to imagine that none of those answers are actually optimized. So I think at some point in the future, let's take 10 years as a benchmark, virtually every protein therapeutic is going to be generated and not discovered with the heuristics and either random biological basis for discovery or variations on those themes that we use today. And we hope that Generate is going to be one of the important companies that makes that possible.
Interesting. So 10 years, did you say? Was it 10 years, the prediction that they'll all be...
It depends on where you're drawing the line. So if you're drawing the line on all commercial products will be generated, that's impossible in 10 years. It just takes too long.
Yeah.
I think 10 years from now, it will... discovery will have shifted emphatically to this domain.
Mm-hmm. Okay. We do want to know the size of the computer models that Generate developed in-house. How much did they cost to train? I mean, do you have AlphaFold in-house? Do you have your own AlphaFold that you had before AlphaFold was AlphaFold? Tell us something about the scope and the cost of the models.
Question. So I get this question many times because, of course, there's a sort of check writing competition taking place in the human large language model domain. And one of the challenges of biology is there's a remarkable paucity of data contrasted with quantities of data that human internet and all human language knowledge or visual knowledge represents to date. So these are not billion-dollar-plus models to train. You can train a high-quality model with on the order of $1 million, utilizing the kind of biological data I described, genome sequences and three-dimensional sequences. AlphaFold has been an amazing advance for the entire field, but it's important to distinguish between what AlphaFold allows, which I'll use the analogy is like reading what DNA is saying, at the level of resolving what structures exist within genomes. That is distinct from resolving quantitative function or from going the other direction, which is to start with either an abstract human language or a quantitative statement of what would the DNA sequence be of a protein that can achieve this functional feat. For example, an antibody binding at a specific location. You can't do that with AlphaFold. But there are analogous underpinnings with the tools that we've created. They bypass the use of first principle physics-based models and really ushered in the era of machines.
We have a question about longevity and aging, which I'm going to get to, but first I need to ask a question to set the stage. Some of the AIs we're familiar with, they played chess, they played Go, right? Why did people start with games? Well, the rules are known, and the objective is known. Like you can judge how well the machine did, right? Yeah. Because you won or not won. Now, you're optimizing for binding, right? A drug needs to stick to something so that it turns it off or turns it on. But we're not really, binding is not the objective. The objective is to cure Alzheimer's. Mm-hmm. So can you optimize, is there a way to optimize this process for the actual end goal to cure the disease? And also, sort of paired with that, isn't it true that binding doesn't always get you to commercial success?
Yeah. So we're starting with variations on where generating a binding event allows you to create an amazing therapeutic. And fortunately, there's more than a small number of those. But if you zoom out to all human misery, we don't understand most of that right now. And in fact, one of the biggest reasons that medicine is so challenging is that the causal basis for the diseases that are most prevalent in society have been really elusive. And some of the more abundant data sets like human genomes annotated with clinical phenotype have only sort of given us, very crude outlines of that basis. So though it is distinct from the way that I described us applying our generative models right now, I'm equally confident that machines are going to be the way of, with greater and greater and greater resolution, understanding what buttons need to be pushed to make a therapeutic difference in medicine.
Yeah. You mentioned that the product that is the first, you think, generative AI antibody to get into the clinic. First of all, antibodies, if you look at the list of the top 10 best-selling drugs, I think 7 of them are antibodies. So you have the first one in the clinic. It's for SARS coronavirus 2. It is a novel target, but that would have been more useful to have in 2020, right? Like, I'm not sure. I'm just saying it's great that you have the program that can give you this antibody, but the market, I'm not sure that the market requires this antibody anymore, right? We don't have our masks on. Our immune systems have evolved. So it doesn't solve the problem in the market. I mean, is this a test case drug, or do you actually think that the world needs this antibody for people who are infected with SARS coronavirus 2, when there happen to be several that exist that were discovered experimentally?
Yeah, well, actually, the ones that were discovered experimentally also hit those rapidly changing portions of the ACE2 protein. Yeah. So, in fact, Mother Nature has been, of course, a voracious enemy. She's an amazing friend, but she's a cruel enemy. And that includes evading our immune system's pre-existing antibodies for first exposure, upon second exposure, third exposure. And she's been able to evade virtually all of the approved or emergency use authorization COVID antibodies. So I don't know. I mean, it's really pleasing to imagine that the pandemic is in the past and that it won't come back. But I wouldn't bet on that. And we felt like it was an imperative that, despite timing not being perfect, of us having these models, 18 months prior to the pandemic beginning, and then, within 3 hours of the COVID genome being available, we could have started generating antibodies against it. We felt like it was the right thing to do to tackle the potential for antibodies to lock in a conformation of the spike protein, a portion of the spike protein's conformation that we don't have tools for right now. And I hope we never have to use it, but right. I don't know if that's the right bet.
I'm just thinking of cases in which this solution to some binding is not really the answer altogether to the disease. Also HIV, right? We have lots of drugs that stick to the HIV genome and mess it up. And yet it's not the cure for HIV for reasons of biology. The virus hides out. It attacks the cells that are meant to clear it. Have you guys thought about HIV as like a difficult problem? Can AI address?
We haven't done anything in HIV,
okay.
Although it would be really interesting. But you're right. I mean, antibodies are just one component of protein therapeutics. Like, this is going to be the way that all protein therapeutics are created. Right. They're just part of the reason it has not been fun to be a human biologist thus far is biology just doesn't work in ways that our brains are well suited to understand, or that chalkbooks and textbooks make wonderful sense of. And I don't think we'll learn it the way that the human large language models have been created, because so much of the narrative we've created about how biology really works is just that. Right. Some fraction of it is wrong. So 50+% of approved therapeutics on a rolling basis now are biological drugs.
Right.
And we've gained conviction that this provides advantages when a biological drug like an antibody is the right approach to be taking.
Okay. I want to drag you into a couple of the flashpoints of the AI debate. First is hallucinations. What if your drug is a hallucination? Then what? How do you know? And then the second is to get to the existential risk question. People are debating existential risk, even at MIT. Some professors have signed on, says there needs to be a pause, stop working on AI for 6 months. I doubt that occurred at Generate. So Do you guys have a role in this existential risk debate? Are you contributors to existential risk in some way? So hallucinations and risk.
Yeah, great questions. I'll answer them in that order, but it's tempting to do the opposite. So hallucinations are sort of a fascinating domain in that there's a version of hallucination which is creativity. And if you're directing models not towards regurgitating existing human knowledge, but towards blazing new trails of knowledge, then creativity can be a virtue. And that line between when, creativity becomes hallucination, becomes unproductive departure from reality, arguably is like the very essence of the scientific mind's, shining light on areas that are beyond our current state of definitive knowledge. So when one is validating, verifying, assessing the merits of any given suggestion, that blur of creativity towards hallucination may be a productive thing. When you have a bedside AI that is making decisions that are followed in a rote fashion without, $50 million of preclinical validation and toxicity testing and, the requisite steps for FDA scrutiny, then of course it's dangerous. But the longer we've done this, the less sure I am about where one draws those boundaries for these types of models when scientific validation is a part of the process.
Right. Getting to existential risk, do you think there is one? Are you part of the discussion? And are you causing it?
We are not causing existential risk. Feels like an appropriate thing to say. We aren't synthesizing anything like a life form. Like everything we're doing is a DNA sequence that is the binding domain of an antibody, a protein on its own. But I don't think anybody should feel comfortable in this century with where biological tools are going to go. And I don't feel comfortable with where they have been. Like, if SARS-CoV-2 is a test missile of what a biological weapon might need to look like, you don't need any generative AI or any of the advances of AI flowing into the process to have real concerns. And so safety, oversight, engagement with publication, engagement with regulators is it's going to have to be a really, really big part of how the field advances.
Okay. Geoff, thanks a lot.
Yeah, my pleasure. Thank you.
Terrific.
Thank you all.